When do they find out? Like next week or something?
I think in 2 weeks. We can't announce it until I think the 17th, but yeah, so that's good. How are you?
Okay. Most of my updates are not for this group, obviously, but I'm pretty good. I have just a whole lot that's cooking right now, and like I wrote out a bunch of them yesterday, and it was just like I am going to try to finish something. I'm going to try to put something together for that interest group. Yeah. But the application is a little bit different than I realized. So I don't know. It kind of was like, oh, who would you want to be like on a panel or a to like be part of the people with you? And I was thinking about obviously putting you with that, but I don't know if I should sort of like potentially include some other people who might be adjacently interested in it outside of the lab or not. Do you have any thoughts about that?
Well, I think, yeah, I don't know if there are any. I can't think of anyone like outside the lab who might be interested. You could put a couple people within the lab you might think are interested too. But I don't know. Yeah, I don't know anyone outside the lab who might be interested. It's something I don't know if it's something that like requires like a group of scholars to be working on it for a couple years, or I don't think this one is that. I think it's more like just some idea. In any case, if just put down some people like in the group that we have, and then if there's if it doesn't get accepted, it doesn't get accepted. It's just can't really do anything about that now. So
yeah, I don't. I'm mostly just. I'm a little bit curious. I'm not quite sure what they are. It's a little bit like I looked at it, I heard about it, but then the form itself is a little bit different than what I understood. So I mean, I'm going to go for it. I'll write something up, but we'll see what happens with that. But I mean, outside of that, I still have my own like big things for my own hashtag summer of data are happening. Like a lot of the micromattrix courses are starting up now. And that's just going to be a little layer of stuff, and I'm in the midst of, you saw a little bit of, I don't know if there's any more reaching out. There's talk about the EA, the research thing that Answer might be doing. So there's that, and I just have so many submissions to do and things to write up that about in the group, and there's a lot of things to sort out. Like there's a lot of exciting stuff, and that's just my mindset, and I'll kind of step away. I don't know how much I'll have to say, but I'll be around for this meeting.
Akshay's here. Hello, Akshay.
Hello, Bradley.
Hello.
How are things going for you?
It's fine. I actually found the 3Js very helpful. I'm trying to learn it, and hopefully we'll be able to do it. Like it will be much more interactive than I actually thought. Like instead of Plotly, where we will be just plotting the embryo cells. Instead of that, we can entirely use a framework which was made for this. So I'm excited. I was actually building the landing page still. Like I added some styling and some other stuff. It's not much. I still don't want to share and show you, but yeah, next week I will show the land page, and hopefully some improvements in the segmentation script I did.
Yeah, it sounds good. I was looking at we get all sorts of pull requests, so hopefully this is going to be a good contribution, and I'll talk about.
Where should I send a pull request? Is there a repo for this?
Let's see. What exactly are you submitting?
Mine is like the axolotl embryo 4D reconstruction. So maybe you might need to make one repository or
Yeah,
something like that?
so I think I might make something. I don't know if I have. I have to double check to make sure I have the right repository set up on there. I'm gonna put it onto the DevoLearn platform, so I wanna set that up. I will do that.
Yeah, sure.
All
Yeah,
right. And then so that's. And speaking of which, I'm gonna talk about DevoLearn in a little bit, so it'll be interesting.
yeah.
Yeah, thanks. I just wanted to say to people in India, hang in there. I've been talking to some people, and it's very bad there in terms of COVID right now, and so I hope everyone is doing well. Here's Krishna, and I'll give you my best. Hopefully, things turn around.
Yeah, my mom is a doctor. She faces like much more adversities than us. Actually,
Yeah.
she has to, treat patients like from different backgrounds and different cases every day. It's getting hectic for her too. So, yeah.
So Tharun is here and Krishna is here, and so anyone, Krishna or Tharun, do you have any updates or?
Hi, how are you?
All right, how are you?
Yeah, I'm great. So yes, I want to present something, but I'm running some network issues. I'll present it after 10-15 minutes.
Okay. All right.
Yeah, and I was just on a call with Dick. He said that he'll be joining late. He had to take a shower.
Okay, it's fine. Yeah.
Yeah, we are working on a computer tomographic breast cancer detection model, and I'm kind of studying physics for that. And in the previous year, I have shifted kind of 3 domains from computer science then to bio and now to physics. And now it's all a cocktail in my head.
Yeah. Well, that's good. Yeah, usually, if you're doing stuff like that, if you're doing multi-dimensional, interdisciplinary stuff, or things that involve different disciplines, it's always good to have build one on top of the other, and then you have to put it together. So it takes a while to do that, but.
Yeah. And I read the reviews on the ANN BNN paper, and it was quite, you can say, disheartening. But at least we have a silver lining to the cloud. One of the reviewer, said it could get accepted. But the first one was quite harsh. Did you read the review?
Yeah, I did. It's that's what happened. Yeah, that's normal actually.
Okay.
It was a pretty competitive venue, so it wasn't like they were almost maybe looking for excuses to reject things. And
Hi.
Hello, how are you?
I'm good. How are you?
Good. So we'll talk about it later. We can talk about it on Slack, but right now we're working on other things, so we'll come back to it eventually.
Yeah.
Hello, Shruti.
Yeah.
All right. Did you have anything you wanted to present or talk about today, or?
Not actually today.
Okay.
It's a lot of work things happening right now, exams and submissions, so yeah, I could not.
Yeah. And Tharun, how are you? In the chat here. Fine. All right. So let's get started. Welcome to the meeting. I'm going to share my screen here. So, I hope everyone's doing well. This is what we are going to hear about. Hi, Dick. How are you? Hello? So, I was fielding some inquiries about Summer of Code, and we'll be hearing back. We'll be able to announce people on the 17th of May, I believe. So hang in there. I can't announce anything before that, so good luck. And again, if you don't get selected, I don't know which projects are going to be. Not all the projects might be selected, so there's that. But if you don't get selected, don't worry about it. We can do something, and many people have done that in years past, where they've submitted an application, which was quite good, and then it didn't get selected. But then we worked on something, and then it became something really good. So the first thing, I'm going to touch on a couple things here. There's this Upstream Conference. If you're interested in open source or like maintaining repositories or doing stuff like that, this might be something of interest to you. This is I don't know, I've never been to this, but it might be something worth checking out at least. It's a lot about, they're going to have a lot of information about open source projects and what people are thinking in that space. Talking about community building and founding things and managing projects and software engineering. And so, being a maintainer is something that Mayoka's had some practice doing. And, it's basically where you build an open source project and you need to maintain it in order for it to become successful. It just doesn't become successful right off the bat. You have to put some effort into promoting it, and we're going to talk about that in a minute. But you have to promote it and then maybe accept pull requests and be, sort of meticulous in terms of how things are maintained and updated and put out into the world. So that's what we're trying to do with DevoLearn, and this might be an opportunity to learn more about that. Now, in terms of DevoLearn itself, this is the repository here, once again, and we have DevoLearn, which a lot of you are familiar with, but we also have other repositories in the... Oh. Someone's dog is not happy. So, we have a number of different repositories here, as well as DevoLearn. So we have data science demos, which I think a number of you have submitted to. We have at least 9 contributors to that, and all of the different demos that have been made there. We have a contribution guidelines repository, which actually needs to be updated. I'm not doing good maintenance work on this necessarily, but this is the kind of thing we need to update on a regular basis. I know that Mayoka is also working on another document on sort of a long-term plan for things, and so we'll incorporate that into this as well soon. We have different organisms for DevoZoo, so we have different model organisms, and then I was just asked about where to put things for the Axolotl project. And I'm not exactly sure where that's going to be. I'm going to create a repository for that. And so that's yet to come. So we're building on this, and I just put out a blog post. And I apologize if I didn't mention you in this. I'm just hitting some of the people who've done a lot of different, talking about different things in the contribution stream. So this is a blog post, and I'll put it in the chat here. This is on DevoLearn maintenance, and I'm just giving an update to people on what we've been doing in terms of maintenance. And so this is, I mentioned that Mayoka's been busy maintaining the DevoLearn pre-trained model software. So that's that one repository in the DevoLearn platform. And I've spent time in 2021 here engaging in what they call technology evangelism to advance awareness about the initiative. So actually I presented 2 talks, one at the OSF Unconference in February, and another one just recently at the INCF Assembly. And, this has been about DevoLearn and introducing people to the platform. And, sometimes people don't necessarily see the greatness of it, but, it's a lot of work, but people don't necessarily see what's all there. But at least getting the word out is the key here. So, so the DevoLearn pre-trained model, we'll talk about that. We've made a lot of advances since January, and Google Summer Code is really facilitating this. And then I talk about these talks here. So if you want links to the talks, I think we have some slides here and a video here. And then I talk about the onboarding guide, which is something that we've been developing. Actually, Krishna was involved in that as well. Ujwal actually took ownership of the Basilaria project 2 years ago, and now he's going to be a mentor for that project. And so we talked a little bit about that. So that's just an update on the DevoLearn platform. I wanted to put that in. Akshay says his internet is crashing. He'll be back in a few minutes. So yeah, Akshay was the one who actually asked me about the repository. So thank you for that. So that's a little bit about DevoLearn. Now I wanted to give an update on the Networks conference. So Networks 2021, there was an abstract that was accepted. This Embryo Networks is generative divergent integration. And so I've been working on the slides for that. So I kind of keep these slides where I keep like parts of the abstract that was submitted, up at the top to remind everyone of what the flow should be, or what we should be covering in this. And so this may diverge a little bit from the abstract, but hopefully not too much. I have some slides from previous talks on embryo networks. So I don't know if I've ever presented a whole talk on embryo networks to this group, but to give sort of a rough idea of what they are, here you have a bunch of cells in an embryo, and they have centroids, and each centroid has a distance between them. And so you can characterize distance in an embryo in terms of three-dimensional space, but you can also characterize it in terms of time and context, which is this theta. And context could be any number of things, and it could be multi-dimensional even. So it doesn't need to be this 5-tuple. It can be even longer, where it describes sort of the state of the embryo at any given time or any given spatial location. And so this is what the network looks like. It's just connecting cells, cell centroids, depending on maybe how close or far away they are in space. And then that changes over time, so the network changes its shape over time. And so it relates to the lineage tree in the following way. You have a lineage tree where cells are dividing, and then they're producing these lineage trees. And the cells at any given point in the lineage, and we usually use a distance threshold. So this is the point mass for an based on cell tracking data. So a lot of you who have been working on the DevoLearn project know what cell tracking data is, and it's where you have all these dots representing cells, and those dots are centroids. And so what they do is they sample the centroids, and they build this model. This is over like many, many embryos, just sampling that space, and this is what it looks like. And of course the cells need to be labeled in order to make sense of this cloud. And then what an embryo network is it further sort of sorts that space based on their proximity or based on some other criterion that you can, create an index and then threshold that index, and it determines which cells are connected. So if they're, very close to one another, they're connected. If they're a certain distance apart, they're not connected. Alternatively, you could have like a signaling. You could have a hypothesis say that there's some signaling molecule that diffuses across the embryo. And so based on that diffusion, you can actually use that as a criterion for connectivity as well. It gives you a different network, but it tells you something about what you're trying to understand. Maybe I want to understand how something diffuses through the embryo, or I want to understand the distance between cells in the embryo over time. Do we get like these changes in structure in the network? Do we get like little modules of the network, or does it remain pretty much randomly connected? And so this is what an embryo network looks like. It's one of these hairball things that I think people have seen if they've looked at like papers on like protein-protein interactions or like social networks. You get these hairballs, which are very hard to interpret on their own. But we have statistics, and we have different things like clique analysis that can help us sort through these connections. And there are other ways to do this as well. This is what it looks like if you look at like C. elegans in terms of the lineage tree. So this is one subtree, sublineage tree, where all these cells are connected at different levels of depth. So this is what the network looks like in space. You can see how they're all connected, what the relations are. And then we can look at actually spatial connectivity in the interactome. So this is actually, this is based on a connectome. If you go and you look further into development and you look at how the connectome is connected, connectome based on gap junctions, they have this sort of structure where, they're connected in various ways. Well, these cells have precursors in the embryo. So these are neurons in the connectome, and these are their connectivity based on their sort of ancestral developmental cell lineage. So this is all of the connectome cells in ABAL. These are all the connectome cells in ABAR, ABPR, and ABPL. And it tells you something maybe about, in those sublineages, what kinds of things they're contributing to later on in neural development in the connectome, how the brain is connected. So I talk about cell division in these networks, and then I give an example actually from the connectome. So what I'm trying to do in this talk, and I haven't sorted it out yet, is go back and forth between the connectome and development, the sort of the building of this neuronal network in the worm. So all these cells here highlighted in this orange are these neural cells that are emerging over development. And this is put onto an adult worm to show kind of where they are in the adult. So they're all over the place in the adult. A lot of them are clustered in the head, some are clustered in the tail, and you have neuronal cells that go down, up and down the body line. And you can see, and this is what they look like in the embryo. Their locations as precursors in the embryo. As they're being born, where are they being born? And they're born on these two different axes. So this is something we can use to understand our network a bit better. Finally, there's this principle of genetic divergent integration. So this is the main idea of the talk. You have this example from an 8-cell embryo, and this is just a generic example. This is where you have a bunch of cells that are connected by distance, but there are no neurons in this yet, just developmental cells. And so these developmental cells form this continuous network. And then the 24-cell example, it's a little bit clearer what happens. The network is joined together, but you also have two networks here. You have this neural network that's forming. These green dots are the neurons, and these blue dots are somatic cells or developmental cells that haven't differentiated, and they form a separate network. But these networks are integrated, and that's the whole point, is that these two networks sort of evolve in parallel, but they're also integrated, and we want to see what those look like. And then kind of consider the idea that maybe that networks in embryos and in connectomes, and then combining the two, don't behave like in any typical regime of a complex network. And so, we think of networks as being random, scale-free, or small world. And so these are what these statistical signatures look like. It's possible that these networks follow none of these types of regularity. And so what if the correct model is not a complex, scale-free, or small world network? And so I talk about a couple of different options here from the literature, and then some things about new world networks, which are networks that expand over time and that find, connectivity patterns as they emerge. And then talking about new types of topologies. So this is pretty disorganized right now. I have to work on it some more, but that's the basic idea of the talk. I'm gonna work on it some more and bring up. I'm gonna maybe try to do some data analysis for this, or build some more animations. I was working on that one figure for a couple hours. It was a little difficult to figure out how to put some of these to visualize some of these things, because it's a pretty hard topic to get your head around, and I want to make sure people can do that. So that's something that's coming up. That's due, I think, at the end of this month. So the conference, I think, is in June. So if you're interested in networks, network science, that's something maybe you should register for and attend. So let's see. Jesse asked a question: is the generative divergent integration slides open to contribution? Are they pretty much set now? Well, I mean, we can. I guess I can open them up to contribution. Why don't I give the link in the chat, and we'll follow the same principle that we usually do. If you have some comments, you can leave a comment. Don't try not to change the slides per se, but you can leave comments in them, and then we can review them. Yeah. So I mean, that's something that'll probably become a paper at some point too. I'm just doing a presentation now because I think it's something that I presented in 2017 at NetSci, which was the networks conference precursor when they were live in Indianapolis, Indiana, and they were sponsored by Indiana University, which they do a lot of network stuff, but it's mostly social networks and other types of networks that aren't what I'm talking about here. So I did a like a satellite session and a regular session talk, and it was pretty interesting. It was fairly well received. So this is a topic I think that, and I've talked about this topic with other people. I think this is an interesting topic that might have some sort of legs to it. So I definitely think we should pursue a paper on this topic eventually. And then, of course, we have other network interests. So we've talked about, like with our submission of the artificial neural networks and biological neural networks paper, there's some talk about networks in that. Oh, note another use of connectron. I'm going to make a note of that. Oh, you have some papers on connectron. Oh, right. So here's some papers on connectrons if you're interested. I think this is... yeah, it's a similar idea to complex networks. Well, actually, complex networks is based on graph theory, so it's all based on graph theory. But if you go to a complex networks conference, they don't talk a lot about graph theory, so they're not trying to sort of solve graph coloring conjectures or anything like that. It's a highly applied audience, so they have certain research topics, their measurement techniques and all these other things. But it's a pretty interesting experience. So I don't know if I don't know if Shruti was going to present or Krishna was going to present. I think Krishna said he needed a little bit of time to get his stuff ready.
Hello.
Hi, how are you?
Yeah, actually, whenever I try to connect with my laptop, there is some error.
Okay.
Just give me. Yeah, you can continue once I'm ready. I'll let you know.
Okay. Why don't we go over the submissions and then we'll... Oh, okay. I think... You're not ready. Okay. We'll go over the submissions and then maybe we'll... Actually, let me unshare my screen. Do you need me to do that or...
Hello, can you hear me?
Yeah.
Is there any disturbance or it's fine?
Oh, no. Okay, try it now. I unshared my screen.
Okay. So, you can hear me clearly, right?
Yes. There we go.
Okay. So, is my screen visible?
Yeah.
Okay, hello everyone. I am going to present about salamander, also known as Axolotl, and it is also known as Mexican walking fish, and it's related to tiger salamander. And reasons to study about them, because their limb generation and evolution. The feature that attracts most attention is the healing ability. They can heal without any scarring and capable of regeneration of entire lost appendages in a period of months. And in certain cases, most of the vital structures can also be, regenerated by them, and that is kind of a great research issue, because if we are able to mimic their regeneration, we can also humans can also regenerate their uh, wounded parts. So, second reason that the 32 billion base pair long sequence of the axolotl genome was published in the year 2018, and it is the largest animal genome completed so far. It reveals species specific genetic pathways that may be responsible for limb generation. So, it has, you can say, it has the most database of not wholly complete, but the best possible long sequence of genome in all of the animals. So it has a lot of things that can be, deducted from them. A lot of research can go on here, and the top of his embryo is transparent, which can expand direct capture of brain development, making it a key representative for molecular investigation. Unlike most of the embryos, its embryos is kind of translucent, so one can, it's kind of visible to see. It's kind of like an house with an open window where we can get glimpse of what is going on. And the most and a great reason that they are so cute, and another reason to study about them. And the data image that I got from Bradley was something like this. And I kind of used Python programming language and converted the static JPEG format into dynamic graphic interchange format. So one can find glimpse about their movement from there, that how the development is happening. Since the image was not of very good resolution, so you can see that there can be a little noise that can be somehow improved by super resolution or something, some other techniques. But instead of, looking to static images, we can find a pattern from here, that how the things are going on. So it's kind of a video made from images in Turing language. And these are the references, Susan's paper and Dick's paper and Wikipedia. So that's it.
Good. When you go back to the slides, I want to make a couple comments here. So,
Sure.
yeah. So first of all, go up to the first one. DevoWorm is misspelled, but
Oh, alright.
yeah, it's okay. But more importantly, so like in the first couple slides, you mentioned salamanders, axolotls, and so axolotls are a type of salamander. It's a species. And then, sometimes in biology talks, they give like the sort of the taxonomic sort of position. So they show like a tree of life, or they kind of make very specific to where it is in the tree of life. So
Are
that's
the trees of life are something like phylogenetic trees?
yeah. So like if you've seen the trees in some of the papers we've reviewed, usually if you look up like tree of life axolotl on Google, you can find like or whatever search engine you use, you can find like some information, and then so that's one thing usually people do. But yeah, I think that's a nice little introduction to it. So I had a couple questions in the chat. Dix points out that in the regeneration slides, you talk about regenerative capacity. Then he also points out that they also study heart. It's a model of heart regeneration. So limb regeneration and heart regeneration. And those are, I don't know. You might be able to find some images of some experiments where they do this. So, I mean, if it's something that you want to really kind of demonstrate to the audience, a really good thing would be to show some pictures of like a regenerating limb, or also spinal cord. So limbs, hearts, spinal cords, those are all things that they study. The reason they study axolotl was, in fact, a lot of amphibians have this capacity for regeneration, but I think axolotls are particularly good at it. I know that they also do some things with newts as well.
They have one more thing that they don't leave scar when they regenerate. It's like it's kind of good as new.
Yeah,
so that's one more thing. And thanks for the suggestions. I'll get back to a data set that kind of demonstrates the regeneration, and I'll work on that.
Yeah. So let's see. Dick says he posed a challenge. Can we read the differentiation tree of the axolotl from its DNA sequence? And it's a challenge that if we, could find the right data set, we might be able to do that. But that's something we've talked about in the past. Then Akshay asks, did you morph the images to make a video? So...
hello?
Yeah, you hear him.
Yeah, I just got a little bit into cropping because a lot of area was, white and doesn't had any, any importance. So I not exactly morph. I just kind of remove this portion in the graphical form.
Cool. So actually, there are so many images, right, in the dataset provided. I think about 2,000 or 3,000 yearly images.
Yeah, lot of images. I took 12, 13 images.
Yeah. So what my recommendation would be is maybe write a script which does that. Like if you are gonna manually do it, it's gonna take a lot of time.
Yeah, I'm not doing it manually. I just had to give the regular expect to my script. For example, this all
We're losing your audio, Krishna.
Yeah. I wasn't able to hear.
Yeah, I can't really hear you very well. I don't know. Okay.
Your audio is...
Okay, I think he's gonna come back in. But yeah, that was and so yeah, Jesse said good intro. Thank you. So yeah, I don't know what we were. Okay, he's back.
Hello. Yeah, I reconnected. Can you hear me now?
Yeah, better.
Yeah.
Okay. Yeah. So I was saying that I didn't do them manually. I used regular expressions that are done for tasks like this when you have to select a lot of things and all of them have a pattern. And for example, all of the images end with .jpeg or PNG or JPG, whatever the expression is. So I just have to give a regular expression to my Python script, and it's like order in the order.
Okay.
Okay.
Have you got the Python? I would like to take a look.
Sure. I'll send them in the GitHub and I'll share the slides.
Alright.
I'll just share the repo link. It's better, right? Share this link.
Sure.
Yeah.
I have one question. Bradley, have you ever seen axolotl in real life?
Real life? I think once or twice. Dick knows a lot more about them in terms of like working with them than I do.
I have a question. Dick, can we pet them? Do they make good pets? Because I find them very beautiful.
So Dick says axolotl is similar to our common ancestor. So the idea would be the common ancestor would have been able to regenerate as well. He also says they're easy to maintain in an aquarium and breed. So they have breeding colonies of them, and they use them to get the they're nearly extinct in the wild. So they're from central Mexico, pretty much. That's where you can find them. And yeah.
While I was searching about them, the axolotl and jellyfish, they can be a key for generation and long life, because they both kind of have this ability to regenerate.
Yeah. He said that they have hobbyists have a lot of breeding colonies and maintain them and so forth. So
Okay.
yeah, they're
Thank you,
nearly
everyone.
extinct in their home range.
Okay. Thanks for the suggestions. I'll get back to them. Thank you.
Yeah. So yeah. Yes, it is. So did Shruti have something to present, or was that something I was assuming?
No, today I don't have anything.
Okay, thanks. Okay, yeah. So yeah, next thing I wanted to talk about was this. I'll get to the papers in a little bit. I want to talk about submissions. Ah, Susan's here. Hello, Susan.
Hello, can you hear me?
Yeah.
Okay, I'm writing an exam today, so
Right.
that's why I haven't joined, and I've been messing around with that.
Oh, that's fine. Yeah. Welcome. So I wanted to go to the submissions document. I just wanted to make sure that we were up on the latest deadlines. Not much has changed in the last week or so. I'm still working on a number of things. The non-neuronal cognition submission is still in progress. The DevoLearn paper is still sort of long-suffering, but also in progress. Mathematics of DevoWorm is still outstanding. So I know I presented on that a while back now, but we're still kind of thinking about that. There's this Williamson Symbiosis. So this is a test of an idea that different organisms, they sort of retain developmental programs from different organisms. So you have organisms that do things like metamorphosis, and Williamson proposed, I believe, and Dick, correct me if I'm wrong on this, but that he proposed that those organisms have multiple developmental programs that overlap and that get expressed in different parts of development. So the idea would be to look at maybe some genetic data to see if we can find evidence of this or some other similar signature. So that's quite hard. And if people are interested in bioinformatics, if they're really into that, this might be something you would want to explore. We also have these molecular level simulations of diatoms. So a lot of the stuff we've talked about in the group was segmenting images, creating dynamical simulations or dynamical representations of movement and things like that with using machine learning. But this would be actually simulations at the molecular level of these movement dynamics or the things that are responsible for movement. So that's another thing that's outstanding. Other than that, we have a number of things that are out in terms of being submitted. There was the artificial neural networks, biological neural networks paper that was rejected by AWIFE, and I talked to Krishna earlier in the meeting, and he was a little disheartened by the reviews, but I think that it's not so bad. I looked at them as well, and reviews often look worse than they are. So sometimes people are very critical, and you just have to power through and figure out how to remedy if you're trying to submit it to the same venue as a revision, it's a little bit harder. But I think if you just take the grain of what they're saying, you can. Sometimes people will kind of make things up in the review. They'll have a certain idea of what the paper's about, and that's not what the paper's about. So you have to balance that out. But I think it's valuable for people to learn how to respond to reviewers and understand that some people are going to see your paper differently, and you have to just kind of make sure it's broad enough so that people can understand what you're saying, but you can't please everyone at the same time. That's the idea. And so, I think we can revisit that paper and come back to it later. So Dick said that okay, so this is contact information if you want to order axolotls and eggs. It's the University of Kentucky. I think they probably want you to do do research with them or something. Not just I don't know if they'll just hand them out to you, but that's if you want to inquire about using axolotls as a model organism. And then yes, then they are due to successful matings between different species.
I know a fair amount about trying to acquire axolotls, and I get quite a few from Kijiji in Canada, at least.
Okay.
And what you do is you keep the males and females separate, and then if you want them to mate, you make some cold water and dump them in it, and that often works.
Okay. Yeah. All sorts of tricks for culturing stuff and establishing breeding colonies. Yeah, it's pretty tricky. So
there's a novel where that works on people too.
Yeah. So I think the statement, and they are due to successful matings between different species, was.
Yeah, you got to Williamson.
Okay, yeah, Williamson. And then I am writing a paper in Mathematica Code for Rafe simulation of the kinematics of diatom motility at the molecular level. So this is the submission that is what Dick is working on, and then this item in the submissions, this number 24, is due to this. I'll put this in details here. Submission. And then, so yeah, if you're interested in that, we can talk about that offline. And then, there's a conference opportunity here. Conference on Complex Systems. Abstracts are due May 20th. NeurIPS, again, is May 19th, or the abstracts. May 26th is the full paper. And then they'll be having satellite sessions of NeurIPS where the deadline is later. So if you're interested in some sort of machine learning or AI stuff, NeurIPS, maybe one of the satellite conferences might be better. Not necessarily the main track because that's quite competitive, but and then the conference on complex systems might also be a candidate for some work as well. But we'll stay on top of that. So that's all I wanted to talk about with respect to submissions. Finally, I wanted to talk about some papers. The first one I wanted to talk about is this paper, Branch Specialization. This is something I've been wanting to get to for a while. So this is from Distill, and this is, of course, the Machine Learning Journal. And we talked about neural cellular automata a while back, and that was a Distill article. And one of the things they have about Distill articles now is they have these threads. And so this is part of the circuits thread. And so this is a format where they get short articles and critical commentary about some aspect of neural networks. So this is part of a collection, and one of these topics is branch specialization. So what is branch specialization? So they say if we think of interpretability as kind of an anatomy of neural networks. So we talked earlier in the meeting about the anatomy of connectomes, and in development especially. This is the anatomy of neural networks. So this would be a neural network and understanding the anatomy, as it were. Most of the circuits thread involves studying tiny little veins, looking at the small scale of individual neurons and how they connect. However, there are many natural questions that small-scale approaches don't address. And so this branch specialization is sort of, I think they're trying to get at is, they're trying to take the idea of biological branching and biological sort of networks based on branching and bring it into neural networks. So they talk about the most prominent abstractions in biological anatomy involve larger scale structures. So, for example, individual organs like the heart and circulatory system, or entire organ systems like the respiratory system, where you have a lot of branching in, say, the lung, and then it's taking in or pushing out, things from the rest of the body or from the circulatory system. And so, these systems are very broad, but they have these branching patterns that allow it to maintain some sort of structure and transport things across the network. And so they wonder, is there a respiratory system or heart or brain region of an artificial neural network? Do neural networks have any emergent structures that we could study that are larger scale than circuits? So this is a question of like these huge emergent structures that we're kind of interested in this group in terms of like biology, but they're actually talking about neural networks. So they describe here branch specialization, or one of three larger structural phenomena we've been able to observe in neural networks. So the other two are called equivariance and weight banding. And those are separate articles. So you can see here, there's the article on weight banding is next. So I didn't look those over before the meeting, but you can look at them later. So branch specialization occurs when neural network layers are split up into branches. So each layer, of course, is, a lot of times we think of it as this homogeneous thing. But if you split it up into branches or separate streams, then this is when you get this sort of branch specialization. The neurons and circuits tend to self-organize, clumping related functions into each branch and forming larger functional units, a kind of neural network brain region. We find evidence that these structures implicitly exist in neural networks without branches, and that branches are simply reifying structures that otherwise exist. In other words, if you take a neural network and you run the simulation and you get connection weights, those connection weights will tend to segregate into streams or branches. And so you'll see this in the brain with different functional regions where, you don't just compute on everything in the brain. There's certain functional regions for certain things like vision or for learning and memory or for attention. And this is just in the human brain, mind you. But those areas are specialized, and it doesn't just, run, the signal doesn't run all over the network. It's specialized into these streams. And so you see this with respect to like river networks as well, where you have, energetic, optimization in river networks where you get like, you go from little streams to tributaries to rivers, and then finally to the river delta. And there's a transfer of energy into sort of the maximal expression at the delta. And so you get this network of, this energetic network, and then productivity follows that. And all these things happen because it's basically based on this network structure. So that's what they're getting at in this article. And it's really good because I think it kind of lays out this idea of branching and sort of the heterogeneity of networks. So you can see again, you have a couple of AlexNet, for example, features some of these branches. Inception version 1 has different branches called inception blocks. Residual networks are not, I guess this is a special type of network that can, they have these sort of pseudo branches that branch off. And so there are a lot of different structures you can implement in your neural network. And so why does branch specialization occur? And so the reason why this occurs is because you have it's defined by features organizing between branches. So in a normal layer, features are organized randomly. A given feature is just as likely to be any neuron in a layer. But in a branch layer, we can often see features of a given type cluster to one branch. So this both reinforces this branching mechanism, and it's almost fractal. In other words, it sort of reinforces additional branching structures down the line. And you have this, they think that there's some role for positive feedback during training. So there's a positive feedback loop that reinforces these branches over time. And so the more data you add in, the more these get reinforced. It's like a river, a branch of a river that, gets reinforced maybe by, the flow of water, or like a trail system in a forest where people walk through and they clear the area. And if people keep going using this certain path, it becomes progressively cleared and it becomes the path of choice. Things like that. There's a reinforcement mechanism at work. And then they talk about branch specialization beyond the first layer. So they just show an example of one layer. Now they're showing it across the network. And why is it consistent? And they give some technical details here. And then they talk about parallels to neuroscience. So, of course, this is of interest to the ANN, BNN paper. They talk about it being observed both in neural networks and in artificial neural networks. And they hesitate to claim explicit parallels to neuroscience, but it's tempting to draw analogies. And so they have this consistent specialization idea where you observe this in the brain, and they think it maybe has the same mechanism. But of course, in biological brains, these mechanisms are reinforced by evolution and development. And so they don't want to say anything outlandish, but I think it's a good article to go over. And they have some comments too. So this is a nice article to read, to follow up on, or to read. And so let's see what we have here in the chat. We have a couple of things. Jesse said, might you want to mention the Bio-Inspired Cognitive Architectures Conference from Oral? Okay, so there's this, and I can send maybe next week. I don't have the document open, but there's this conference going on. It's called Bio-Inspired Cognitive Architectures. And so they're accepting papers on neural networks and AI systems that might be of interest to some of you. Maybe if you could post an announcement of it in the Slack, Jesse, in the DevoWorm Slack channel, that would be good. And they're accepting both abstracts and papers, and the deadline is a ways off. So if you're interested, you might want to check that out and submit something. Dick says growing graphs. You see Stephen Wolfram. So this is about growing graphs. This is a YouTube video from Stephen Wolfram on this topic. So this is something when we talked about earlier in the meeting about like how graphs, you have these graphs that grow in terms of their topology. How do you characterize them? What are their organizing principles? How do they process information? So it's something else to check out. And I listed the distill link here. So I'm going to finish up the meeting by going over a couple papers. If you have to leave, that's fine. But I wanted to go over a couple of things here. So we have a lot of things in the queue here. I think Dick sent me this last week. This is a classic paper from Waddington, Conrad Waddington, who is known for his epigenetic landscapes idea, and Russell Coe, who is someone else I'm not familiar with, but he's a computer scientist, and Waddington was a geneticist, and they put this paper out in 1969 called A Computer Simulation of a Molluscan Pigmentation Pattern. And so this goes back to the things you were talking about with the seashells and the patterns on seashells. So Shruti, of course, is interested in that. So the idea is that they're interested in interpreting like seashell patterns and analyzing it with a computer to see if they can find, figure out how these patterns emerge on the shells, because, they have some interesting mathematical properties. We want to understand morphogenesis, how they form, because they look very organized as structures. And so we want to understand how that works. And so they ended up using a program. So the formal characteristics of a color pattern on the complex appearance on a molluscan shell. So if you've seen a seashell, you know what that looks like. We've seen those. Were studied by developing a computer program which produces an acceptable simulation of it. So they didn't really use anything fancy here, especially in 1969. The pattern clearly involves a random factor determining points of initiation of diverging lines of pigment. The program contained parameters controlling the density of random initiation points in different sectors of the growing edge of the shell, and the angle of the divergence of the lines. So they had to set this problem up in some way. And they used a certain approach, and it's in the paper. In order to produce a satisfactory simulation, it was necessary to introduce another rule, by which new initiation points arising within a certain distance of an existing pigment line are drawn onto that line. It has shown that formal rules of the program can be given a plausible physiological interpretation. So what they did was they used a couple of rules in tandem to develop these pigments. And so, there's a sort of contingency where the rules had to be in place and they were able to produce this pattern. So this is something, if you're looking at like cellular automata, you'd be interested in knowing what the rules are so that you can reproduce them. With machine learning, though, we may not need rules per se. We just need some exemplars to sort of, train the model on, and then look at the model and see how it's interpreting the patterns inside. So, there are different ways you can do this. And this, of course, was back in 1969, so they were really using rule-based models. So it was very much, but it very much gets you down to sort of the nitty-gritty of the problem and how to set the problem up. So I think that's why it's worth revisiting some of the old papers, because they actually do talk about this very much like the way they set the problem up. And so they talk about the Turing model of morphogenesis, which is sort of, let's see what they say about that. So the pattern that they observe differs from those studied by Turing and Scriven by being diachronic in the sense of Waddington. That is to say, the shell does not exhibit the instantaneous state of the pattern-forming system at a particular moment in time, okay, but rather the pattern that is built up gradually over a period of deposition of pigment at the growing edge of the shell. So what happens is you have this, basically the spatial system that goes from one end to the other, and you have like this thing that's laid down over time, and there's a deposition. So it's almost like a printed document that comes out of your printer. It starts printing from the top and goes down to the bottom, and it might go back over for different colors, and there's a bunch of stuff that's deposited on the surface, and then that's your paper. And you say, well, how did this paper get printed? And so the answer is there was a printer head that went over it and printed it. And in this case, it's more about like, it's something that is diachronic. It happens at different points in time, and it's just the accumulation of all of that. So this is building up gradually, and whereas the Turing model is more like it deals more with the interactions of morphogens. It doesn't necessarily depend on the deposition of things over time. So that's the difference here. So it's a different model of morphogenesis. And so, again, depending on how you set this problem up, you get a different result. So this is an example of a shell that they're looking at, different shell pictures. And then this is their diagram of their 3 stages. This is their morphogenetic model. So this is where you get this, in time, this is how the pigment is deposited. So over time it gets deposited, and you end up at the end of the simulation with these patterns, like some patterns that look like this. So that's what we're dealing with here. So they talk about the physiological model and some other things in here. And here are 2 patterns that result from the first program. So this is not a cellular automata. This is just simply lines that they've looked at that are deposited on the surface, and they wanted to look and see how you would produce something like that. So you can do this without any sort of fancy, model. This is just like a program with rules, and they're being implemented in a certain way. So that's for that paper. The next one is this Reconsidering the Evidence for Learning in Single Cells. And I know that I posted this in the Slack, and then Krishna posted a story about this article. This is Randy Gallistel, who's a learning and memory person, and there Samuel Gershman, who's a neuroscientist. So a number of people who are interested in cognitive science and psychology, and I think Brains, Minds, and Machines, which is a sort of an interdisciplinary group. And they're interested in this idea of learning in single cells. So there, in the early 20th century, there was a question about whether single cells could learn. And so learning means like conditioned learning, the type of very simple learning that maybe we observe in sea slugs we talked about, I think, last week or the week before. And they do this thing where, it's like they associate different stimuli, they do things like that. But of course, in single cell organisms, they don't really have brains. But they can do this sort of thing, nevertheless. And so why is that? And so we've talked about non-neuronal cognition, and this is basically what they're getting at here. So you can do this sort of thing where there was a view that prevailed that they were capable, that these single-cell organisms were capable of non-associative learning, but not of associative learning, such as Pavlovian conditioning. So this is where you associate like two stimuli. In the case of Pavlovian conditioning, it was ringing a bell and a dog salivating, thinking that it was time for dinner. Experiments indicating the contrary were considered either non-reproducible or subject to more acceptable interpretations. So there was this whole literature on paramecia, which are a group of single-cell organisms that were studied a long time ago by Beatrice Gelber, and they were kind of lost to history. And so people criticized it heavily when it was published, and so it wasn't really accepted. Now people are reinterpreting this as, maybe that they were doing more than what we thought they were doing. So this prevailing orthodoxy was that single cells cannot learn. And so what we found from the work on paramecium, revisiting this work and reinterpreting it, was in fact they can learn, and they can even do associative learning. And so I think this is both an interesting historical aside, but also a good point in favor of this idea of non-neuronal cognition. And so the emergence of learning is much faster than evolution, so you can learn things much faster, and then you can evolve as an organism, and that's good for survival. The focus on learning has led to a biological model of learning based on synaptic plasticity, which of course is good if you have synapses and you have a brain. But if you don't have synapses in a brain, then can you even learn? Well, we see from some of the experimental data that there is learning going on in these systems. So why? And so that's what they talk about here. It's interesting to note that some of the in sponges they found that there are analogous or paralogs to synaptic genes. So like there are genes in sponges that are being expressed in sponges that have sort of a parallel with what's going on in the synapses of brains. And those genes are being expressed in similar ways. They don't get expressed in the synapse, but they get expressed in the sponge morphology. And so that's an interesting parallel. I remember this paper from a while back, but basically they found that there are a lot of analogs to organisms that don't have brains with, some of the mechanisms of brains. And so that's another interesting aside. So in connectionist models of learning and memory, synaptic conductance is represented by sign connection weights. So this is our typical inspiration for artificial neural networks. But in unicellular organisms, we don't have any of this, but they're still capable of complex behaviors. So in this paper, they kind of go through some of the evidence from paramecium, and we don't talk about paramecium much in this group, but so this is the alternative hypothesis for having brains, which is that memory can maybe be stored using a cell intrinsic substrate. So such as RNA, we talked about that with respect to sea slugs, post-translational histone modifications, or DNA methylation patterns, which are actually important in some forms of cell differentiation, and switches that tell a stem cell when to differentiate, or to tell it when to change its state. And so there's a whole literature on stem cells and DNA methylation. These theories posit that posit a mapping from experienced quantities, like the duration of an interval between the onset of a conditioned stimulus and the onset of an unconditioned stimulus, to the hypothesized changes in molecular level structures that encode them. So there's a biochemistry of memory so we don't know exactly what the signals are, but we know they're different sort of event timers that are molecular in nature that might provide interval timing. So they're biochemical mechanisms for the sort of keeping time in the organism and keeping time in terms of what stimuli are coming into the organism. So so this kind of goes through the historical background of what they used to think about these mechanisms at the early part of the 20th century, and then kind of going into the paramecium as a model organism, and then kind of talking about Beatrice Gelber's work, which really kind of brought some of this together. But of course, this wasn't interpreted that way because the peer review process didn't accept those interpretations. And so this is an example of some of the experiments that were done, and then kind of going through this whole intellectual process, but also in terms of what might be happening in single cells. And I think it's an interesting paper. We should definitely revisit this later. Let me see what we have here. Let's see. Jesse says, this is also interesting with respect to Paul Cizik's recent paper. So this is something we covered in my other group. We talked about this idea of, how did the brain evolve? They're different, especially how did different developmental precursors of the brain evolve throughout the tree of life. So we have these different structures in the human brain or in the mammalian brain that this man, Paul Cizik, wrote a paper on looking at this phylogeny. And it wasn't really like a statistically supported phylogeny. It was like they put different brain structures on a tree and, evaluated maybe when they arose in the common ancestor. And so it's an interesting paper. But yeah, it did have a lot to do with it. It's very interesting in that context. And then Dick says, we may have to solve the problem of single-cell morphogenesis before single-cell learning. And that's probably true. There's a lot there in terms of single-cell morphogenesis that we can understand as well. And maybe they're connected. We don't really know. That would be an interesting question. I think finally I'm going to talk about, and I don't really want to get deeply into any of these, but I'll talk about this paper. So the pace of development. And so this is something that, if you're interested, I know we had this paper that we published on timing of cell divisions and cell differentiation in different embryos and what the temporal pattern of that looked like. And this is a news article from, I think, Nature. So this is talking about the pace of development. So researchers are starting to work out why animals develop at different speeds. The key could be tiny timekeepers inside cells. So we just talked about interval timing perhaps being regulated in single cells. And so this is a good follow-up to that because it talks about sort of some of these mechanisms for timing. So when a cell develops, of course, there's this whole internal program that gets expressed, and there's a certain timing to it. So cell division has a timing. There's a very intricate gene regulatory circuit that manages, cell division. So like at different phases of cell division, you have different things that are expressed and then accumulate and then trigger other things to be expressed and accumulate and then degrade. And I'm talking about the gene products here. And all those things have to be timed just right in a single circuit for cell division to occur normally. And so this requires some sort of timekeeping mechanism perhaps. But definitely it requires sort of a temporal cadence. And so one could imagine if you speed this process up, you could have more cell divisions in a shorter period of time. If one extends that out, you could have fewer cell divisions over the same period of time. And so, this is something that is talked about in this article. Animal cells bustle with activity and the pace varies between species. In all observed instances, mouse cells run faster than human cells, which tick faster than whale cells. And by ticking, they mean the metabolism in the cells. So, but this isn't exactly cell division, but it's a similar idea. These differences affect how big an animal gets, how its parts are arranged, and perhaps even now how long it will live. So this is more in line with sort of maybe cell division, but also like what we've talked about heterochrony. as well, which is, do cells proliferate at a faster rate or a slower rate. And that's actually controlled by what they call heterochrony genes. So there's a whole literature on heterochrony and timing and development. Are things slowed down or are things sped up with respect to body growth, to like body length and growth. But those things, ultimately reside in the cells and cell division. So there's this quest to look at the cellular timekeepers that control the speed of development and the speed of how these bodies are built and tissues are built and so forth. And so they're saying that there's a clock in early embryos that beats out a regular rhythm by activating and deactivating genes. This segmentation clock creates repeating body segments such as the vertebrae in our spines. And then this is what they're interested in this lab here in Barcelona. So the segmentation clock, you can study it sort of in early development. So you don't have to observe the organism for the entire lifespan or gestation period. You can just look at sort of early development and get a sense of what this clock is doing. So people have been studying this since the 1990s, and they know it runs about twice as fast in mouse embryos as it does in human embryos. So the speed varies by species, and people are trying to figure out why that is. And so they looked at this in chick embryos. They've looked at waves of gene expression moving along the embryo from tail to head, oscillating in synchrony with the development of somites. So there's this wave activity. Actually, this is something, maybe differentiation waves has something to do with this. I don't know. This is but it's nice to see these things in other parts of developmental biology. Oh, so they do mention Haeckel and heterochrony in this article. So they talk about heterochrony here, and this is at sort of the organismal level instead of the cell division level. But this sort of clock contributes to heterochrony. And so modern developmental biologists regard heterochrony as a key concept that helps to explain a core mystery. At the earliest stages of development, all vertebrate embryos look alike. Yet as the embryos develop, they become easily recognizable. So how do they differentiate in terms of something that can be, from a very generic embryo to something that is like the different organisms that we see, the diversity that we see in the adult form? So how do cells in a human embryo develop into a human baby and not into an infant chimpanzee or juvenile fish? And so some of that is determined by the speed at which the parts of the body develop. And so that can be, if you were to slow that down in a single species, you could get different morphologies. If you could do that, I guess, experimentally. And what they're saying is that there's, if you slow things down, the organism is going to look different if you sped it up. But across species, there's a variable timing that contributes to these differences in form. And so they have this diagram. They show a mouse embryo, they show the somites, and then they show like the tail here, where there's gene expression, and then there are these segments that form, these somites that sort of emerge and expand the tail outward. So it elongates the tail. These somites, depending on how fast they get put in place, determine the length of the tail. And so they, this is a interesting area of research. I know I probably haven't done this justice, but this is a nice paper to go through and to find out more about this phenomenon. So this is just a news article. So with every news article, there are always papers to follow up on different topics. So here's a list of these papers here. So I think that's good. So what do we have in the chat here? So could you send the links to these last two papers here or in Slack? I'll probably put the links in Slack, pace of development, etc. Dick says synchrony and cell division usually just in early embryos. Yeah, so there's differential timing in cell division later on in development. So there's the heterogeneity of of timing and cell division that emerges. But yeah, the synchrony is usually just in early embryos. And Jesse says, I would like to look a bit more into this metabolism with respect to development. Yeah, I think I should maybe assemble some resources, or I can give you some links. And then Dick has a link here, or an article here, duration of cleavage cycles and asymmetry in the direction of cleavage waves prior to gastrulation in Xenopus laevis, which is the frog model organism. And so this is a nice article from 1983 you might want to check out for more information. So anyone have anything to say before we go? Any questions or comments? Okay. Well, I'll follow up on some of these things. I'll send out the papers. If we have anything else that we want to add. Oh, Jesse, you had something to say?
Oh, no, I was just gonna say, good meeting, good to see everybody, and take care.
Yeah. Yeah. Thanks, Susan, for showing up. Thanks, Krishna, for your presentation. Next week, we'll probably talk about more very similar things, but if we have anything to follow up on too, please feel free to contribute or to send papers my way. As you can see, we have a very long queue of papers, but we'll get through them. It's always interesting to see this. So have a good week, everyone. Stay safe. Thank you.
Okay.
Bye.